Let us now talk about the kinds of lysosomes. In different types of cells and at a different times in the same cell, 4 types of lysosomes have been observed. They are primary lysosomes, secondary lysosomes, autophagic vacuoles, and residual bodies. Primary lysosomes. Newly formed lysosomes are called primary lysosomes. They are formed principally as vesicles pinched off from Golgi membranes, but in some cell a specialized portion of endoplasmic reticulum called GERL, that is Golgi associated endoplasmic reticulum, from which lysosomes arise, is probably also involved. Secondary lysosomes. The secondary lysosomes are also known as heterophagosomes or digestive vacuoles. When the cells feed on a foreign or a exogenous or a extracellular substances by the process of phagocytosis or pinocytosis, then the plasma membrane forms the membrane bound vesicles known as pinosomes or phagosomes around these substances. The pinosomes or phagosomes ultimately fuse with the pre-existing primary lysosomes and form the secondary lysosomes or heterophagosomes. The objective of secondary lysosome formation is to mix lysosomal hydrolysis with the ingested material, so that breakdown of the latter can occur. The digestion products like amino acids, nucleotides has a low molecular weight and readily pass through the lysosomal membrane to become sources of energy or raw materials for biosynthetic pathway. As product leave, the lysosome often diminishes in size. This change requires a loss of surface area, which can be achieved by an inward budding of the limiting membrane in much the same way as pinocytic vesicles are formed. The internalized membranes is then digested. This process, which is particularly prominent in the digestion of the pinocytic vesicles, leads to an electron micrographic picture of a vacuole containing a numerous small vesicles and fragments, a structure known as multivesicular body. The fusion of a lysosome with an endocytic vacuole is a selective process. The vacuoles do not fuse with endoplasmic reticulum, mitochondria, etc., nor do the lysosome fuse with a plasma membrane under ordinary circumstances, even though plasma membrane and phagocytic vacuole are made of the same stuff. However, in the presence of cytochalasin B, extracellular release of lysosomal enzymes does occur readily, indicating that perhaps the microfilamentous web beneath the plasma membrane normally protects it from fusion with lysosomes. There is some evidence also for involvement of microtubules in guiding endocytic vacuoles, lysosomes and specific granules, and there is evidence for regulation by cyclic nucleotides, probably via their effect on microtubules. Killing in a secondary lysosomes. Simple enzymatic digestion by hydrolysis is not the only process that occurs in a secondary lysosomes. Many microorganisms, while they are viable, resist attack by hydrolysis. Hence, mechanisms have evolved specifically to kill ingested organisms prior to their breakdown. Lysosomes typically contain lysozyme, an enzyme that degrades bacterial cell wall, especially the simpler walls of gram-positive bacteria. The lysosomes of neutrophils also contain a lactoferrin, which binds iron and other metal ion tightly, thus inhibiting growth of the ingested organisms. The contents of the digestive vacuoles are usually very acidic, that is less than pH 4 in neutrophils, which by itself inhibits the growth of many bacteria. And finally, oxidases thought to be present at the surface of the cell membrane and thus also on the inside surface of digestive vacuoles produce hydrogen peroxide is microbicidal in its own right. But in neutrophils and most macrophages, it is also utilized by an enzyme called myeloperoxidase to covalently fix chloride or iodine onto organic targets such as bacteria. This halogenation greatly facilitates killing of the organism. Autophagic vacuoles. Lysosomes are involved not only in the process of digesting extracellular material known as heterophagy, but also in the digestion of intracellular substances and activity known as autophagy. In autophagy, lysosomes are responsible for turnover of organelles of animal cells, a process that is accelerated during involutional states in a muscle that is not being used, during starvation, after injury, or during the remodeling that occurs with cellular differentiation. Even in unstressed normal cells, the turnover of organelles by autophagy is relatively rapid. The half-life of a liver cell mitochondria, for example, is 5 to 6 days. For peroxisomes, it is 1 to 2 days, and for ribosome about 5 days. The autophagy of cytoplasmic organelles start with an isolation envelope, which is a sac of smooth surfaced membrane that wraps around the material to be removed and separates into a sort of phagocytic vacuole known as an isolation body. The source of the isolation membrane, at least in a damaged liver and kidney, seem to be endoplasmic reticulum. However, work of Michael Locke and his associates in 1975 indicates that the isolation membrane in an insect cell during metamorphosis and in some other animal cells is derived from the Golgi complex. In any case, lysosomes can fuse with the isolation body to create an autophagic digestive vacuole and release a breakdown products to the cytoplasm to be used as an emergency source of food or as a raw material for cellular remodeling. Residual bodies. Although killing and breakdown in the lysosomes are efficient, some materials are simply not digestible. These substances may be released from the cell by fusion of the digestive vacuole with the plasma membrane by exocytosis. This process is very efficient in protozoa, but not so efficient in higher animals. Hence, inactive digestive vacuoles containing debris may accumulate. These debris-filled organelles called residual bodies are of no consequence to cells like macrophages or neutrophils because the cells do not live long enough for significant amounts to accumulate. In cells that do not divide and are not replaced, substantial quantities of these materials may accumulate. In neurons and muscle cells especially, there is a steady increase in the number of these bodies, which are called in this context lipofuscin granules. The age of an animal can often be estimated from their concentration, and residual bodies play important role in the process of aging.